So next, I will show you another genome browser Ensembl. So Ensembl is similar to NCBI. So you want to go to the website of Ensembl, you just type the keyword Ensembl. And then the first one is the Ensembl genome browser. Okay, so so this is what the Ensembl genome browser looks like. The aim of the Ensembl is to provide annotation for the biological community that is freely available and of high quality. So, and the Ensembl database is very informative. It contains the genome sequence, the gene annotation, the gene structure, some regulation elements. So almost you can find all the genome information you need in your project from the Ensembl database. So the names in the Ensembl database is different from what Dr. Tan showed you in the NCBI database. In the NCBI, the transcript ID may begins with NM or NR. However, in the Ensembl, for the gene, the Ensembl ID is begins with ENSG. For the transcript, it begins with ENST, and this is for the protein or the exome. And this is for human. If for other species, so the name begins with this one. Okay. So okay, let's go to the Ensembl browser, and you may explore this gene, or we can see the gene that Dr. show you when using the NCBI database, the IGF1. Okay, you can type IGF1 here, and you can, for the species, you can choose human, and then you click go. Okay, so the first one is the human gene IGF1. Then you click the first one. Okay, so this is for the gene IGF1. This is the name of this gene, and this is the name used previously, and this is the location in the 38 version. Here, if you cannot see this table, you can click this button, and it shows you that this gene has 7 transcripts, and you can see the gene ID is in SG, this number, and for the transcript it is in ST. Okay, so this column shows you the length. Yes, if you put your mouse on the title, it will shows you the meaning of these columns. So this is the transcript length, and this is for the protein. And so this is the CCDS database ID. CCDS is the consensus coding sequence. So if the transcript has this ID, it means this transcript is included in many databases, not just Ensembl, but also NCBI, UCSC, some other databases. So it means that this transcript is a protein coding transcript and is of high quality. And you can see this name begins with NM, so this is the name in the NCBI. And here, this is the flags for each transcript. Similarly, when you put your mouse here, it will show you what does this flag mean. Okay, so when you drag down, and here, this shows you the structure of each transcripts. And here the box refers to axles, and the lines links these two boxes represent the intros. And you can see that some box is white. This means this region is untranslated. And the box with color means this region is the coding axle. Okay, so you can see on the there are two strands. One is the forward strand and the other one is the reverse strand. So you can see this string is located at the reverse strand. You can see from the direction of the arrow before this string name. And also you can see that if the string is located at the reverse strand, then this the string is below this blue bar. And you can see these two low non-coding RNA, the arrow is in this direction, and it means these two non-coding RNA are located at the forward straight. Okay, and those two RNAs are located above this blue bar. And okay, so here you can see there is a gene legend. You can see the transcript are colored by different color, and so the golden one means this transcript is included in both the Ensembl and this database. And if it is just red, and it means this is already included in the Ensembl database. And here, this color shows this is some non-coding, non-protein coding RNAs. And this part shows you the regulatory elements, and such as the promoter. So there are the promoter enhancer. So this is the should be the first axon, and the promoter is near the first axon, and also some other regulation elements like enhancer. Okay, so if you want to know the sequence of this gene, you can go to the side menu on the left side, and then there is a sequence button. Then you click this one, and then it will show you the sequence. Maybe because I configured this page, so it will show you the SNPs. Maybe in your computer it will not show you this information. Okay, so this is if it is colored by orange, this means this is axle, and these letters in gray means this intros. Okay, so if you want to see these SNPs like what I show here, you need to click this one, configure this page. Okay, and so there are many display options. You can ask it to show you more upstream sequence. Okay, so you want to show the variance. So maybe in your computer, the option is no, and then you click show yes and show links. And there may be many variants, so we need to here hide variance by frequency. For the variants that have fewer minor allele frequency, we did not show it. Okay, so just this one and this one. Okay, then you click here. Okay, and then it will show you the location of the variance. Okay. Okay. So if you wanted to see the more details about this variant, you can click this one. Click the SNP name. Okay, so here you will get more information about this SNP. So this is an intergenic variant. It is inside the gene IGF-1. And here is the alleles of this SNP. Some people have allele A at this position. Some people have allele C in this position. Okay, so and this is the location of this SNP in our genome. And click here, you can see the genomic location of this SNP. And here, let's go to here to see the population genetics of this SNP to see which population have this SNP. Okay. So you can see it is said that the ancestral allele of this SNP is A. And here is the 1000 genome project. It includes 1000 human genomes. And you can see, and this is different populations. When you put your mouse here, and it will show you the names of each population. And you can see the above populations, all of them have the allele A for this SNP. You can see. And almost, 100% population have the allele is A. And here is, you can see, this is Chinese, and this is Japan. Okay. And you can see here for this population, and this is South Asian and Indian. And so this population have the allele C for this SNP. So maybe if you got your genome sequenced and you found that in your genome at this SNP position, your allele is C, it means that your ancestor may come from this population. Maybe this SNP is not very convincing, but some SNP may show you where your